Let's move on now to John Chia, who's from UCLA, Los Angeles, ME and chronic enterovirus infection, an update on pathogenesis. John.
Thank you, Ian. And it's really my pleasure and privilege to come back to invest in ME. It's so nice to see so many familiar faces again after 3 years of absence. I think I do have a little more new things to tell you. Well, just a quick review on enteroviruses. Most people are probably not familiar with these viruses. These are small positive strand RNA viruses, and there's 3 polioviruses, 23 Coxsackie A viruses, and 6 Coxsackie B viruses, and 26 echoviruses, and then enteroviruses by numbers. But now the new classification is actually called human enterovirus, HEV, classified as A, B, C, D category. As you know, that last year had a major outbreak of D68. That happened as a respiratory infection in children that led to paralysis. There were about over 1,000 cases, and 150 of the children were actually paralyzed. And the prognosis is still not sure on those patients. Enterovirus infections are quite common. It's probably second only to the common cold. There's 30 to 50 million people probably get this type of infection in the United States per year. This was an estimate for over 15 years ago. Most of the patients may be asymptomatic, but many patients will develop respiratory, gastrointestinal infections with the diseases that you see on the list there. A number of patients can develop prolonged fevers, severe flu-like feelings with intense myalgia. This myalgia is far more than we see with H1N1 influenza. It's truly, truly impressive. Patient can develop leukopenia, bone marrow failure, and this type of infection is much more severe in the immunocompromised patients. These viruses can then spread to the central nervous system, the heart, and also muscles. These, as a group, these viruses are actually the only viruses that can actually go to all 3 sites. Okay, the other viruses can go to 1 or 2 and others. And these viruses has been shown to cause chronic diseases in these organs that I just mentioned. It can involve the genital urinary system and the skin. The difficulty in recognizing enterovirus infections. I am one of these perfect examples. I actually did very well in my infectious disease fellowship, scored very high on my board exam, but I didn't really understand enteroviruses, not until my son got sick. Then I studied. Most patients are not trained to recognize acute enterovirus infections, so they don't actually know what they are looking at when the patient develops an acute infection. If they don't understand the acute infections, they don't really understand the chronic infections. And most patients are not taught, physicians are not taught that enterovirus can actually cause chronic infections. The usual problem with physicians, as you know, if the doctor can't figure out something, then the problem becomes yours. It's all psychosomatic. So recently, we have rapid diagnostic tests, like take a nasopharyngeal swab, and in the patient with acute respiratory illness, we can actually determine they actually have an enterovirus or rhinovirus. Then when the patient develops, of course, this complication of paralysis, those samples get forwarded to the, in our country, state laboratory, then to the CDC for typing. Then they realize this big D68 outbreak. I'd like to review for you very quickly the past evidence for enterovirus persistence in CFS ME patients. Actually, most of these, if not all, came from Britain, and many actually came from the city. So NARN actually found that Rt Pcr assay was a better test than neutralizing antibody for differentiating CFS patients from controls. GO found enterovirus RNA sequence in muscle biopsy specimens from 53% of the CFS patients and 19% of normal controls. Subsequent studies using patients with muscular dystrophy or neuromuscular disease, the percentage was comparable. Cunningham and Archer really found this nice phenomena in 1990. This was a landmark paper that actually established the possibility of double-stranded RNA in the muscle biopsies. There's an even ratio of positive to negative strand, a 1-to-1, rather than the 100-to-1 ratio seen in a control enterovirus culture. There's usually one negative strand, and it can produce hundreds to thousands of positive strands, which would then be translated into protein, and the protein would be cut in pieces, assemble the shell, and the RNA will be incorporated. Then it becomes a virus, a virion. Doche and Ulrich also found the enterovirus RNA in the muscle biopsies, and so did another study. But some of the other studies show negative findings. We Americans have never repeated these studies. Until 2 years ago, Dr. Kim in the University of Nebraska vascular surgery unit actually found 60% of enterovirus from calf biopsies of patients with chronic calf pain and moderate arterial diseases. So the other very important finding was this young lady, 30 years old, who has this dreaded illness for 5 years and committed suicide. And tissues were removed while she was still on life support from the muscles, heart, and various parts of the brain. Enterovirus RNA was found in the muscles, heart, hypothalamus, in the brainstem, and not found in the control samples for one patient with cerebrovascular accident and 4 patients who committed suicide. And the RNA sequence showed 83% similarity to Coxsackie virus 3. This is another landmark study done around here. This was a blinded study of chronic fatigue patients versus normal controls using a 4-primer Rt Pcr and generated 264 base pairs. And the sequence of what they found actually turned out to be enteroviruses. They were able to find the RNA in 41% of the CF serum and also 27% of the CF Buffy coat, which is the white blood cell layer. And control serum, only 2%. That's highly statistically significant. Acute infection, in the stool, they were able to find 28%, but the CF patients, 48%. This was very difficult to reproduce. I don't think any other study have reproduced this finding, but this was the blindest study. So they did the right thing, and this has never been really refuted, but it's never been reproduced. But we Americans actually trying to reproduce these results by sending the specimens to Hardy Robart in Denver from the NIH. And it's unclear at that time how the serum were capped or plasma were capped, how long it took for the people to take care of the blood samples after the blood draw. And at that time, the Federal Express probably wasn't working quite as well as these days. So their finding was completely negative. Okay. I started working on this disease by first finding neutralizing antibody tests for Coxsackie B 1 through 6 and Enterovirus 6, 7, 9, 11, 30 in 1996. That was total serendipity. I'm very glad we were able to do that test. Then I diagnosed my own son's illness in September 1998. His Coxsackie B virus 3 antibody was 640, and Coxsackie B4 antibody was 640. My son had 2 major illnesses, and the second one was probably Coxsackie B4, so a year before I diagnosed him. Normal level would be less than 1 to 10, and my level was less than 10 and 40. So it was clearly different. So at that time, I reviewed the British literature, and then we started working on enterovirus and ME. So the first thing we did was still continue to use the antibody that we draw on patients, since this is commercially available, but only in one laboratory in the whole United States. That's ARUP lab in Salt Lake City. And they use a virus neutralization test, which is quite tedious, but very reproducible. Enterovirus RNA of blood we worked on from 2000 to 2007, stem biopsy from 2005 to now, and skilled mice work from 2010 to 2013. This is the neutralizing antibodies for the patients on the right half, and these are the neutralizing antibodies for Coxsackie B1 through 6, then Enterovirus 6, 7, 9, 11, 30 for the normal controls. 200 patients, 200 controls here, 150 here. You can see the titers. This goes from 0 to 640. There's a big difference here. The difference is black and white. This helped us to at least establish the set point for the antibody titer. So if you're over 160 or 320 for Coxsackie B4, that's a significant antibody titer. So if people say, oh, everybody has these antibody tests, yes, they do. They do. They may have a low test results like this, but they don't have very high results like this. Second thing that we're trying to do after we saw these differences in antibody titers is that antibody titer doesn't really mean that there's still a live virus infection. So we're trying to reproduce the British finding by finding the enterovirus RNA in the blood. We picked the white blood cells instead of the plasma because in the pilot study, we're able to find the evRNA in 30% of the peripheral blood mononuclear cells and only 10% of the serum, and the controls were completely negative. This was a blinded study. But after that, we said we better use a better way to screen it. And this kit actually is made by Chemicon using Hardy-Robar's method. This was the person who tried to replicate the results earlier. But they added another primer to the two-primer system, and that made this test much more sensitive. This test can sometimes get down to 1 copy per well. Most of the time, we get to 10 or 100 copies. So when we did the first study, it was 131 patients. We were able to find the viral RNA in 50 patients. That was 38%. Those are the most symptomatic patients. We kind of picked the patients carefully. And then 2 out of 52 of the controls, or 4%, were positive. And the difference was statistically significant. Then from February 2000 to August 2005, we actually drew more than 2,500 blood samples for more than 510 patients. So we drew 5 or 6 blood samples from each patient and test them again and again. If 2 positive, we call them a positive. So 35%, or 179 out of 510 samples, were positive. This assay is highly dependent on the assay sensitivity. Most of the time, we can get a sensitivity down to 10 to 100 copies per well. So if we had a sensitivity of 1,000, we rarely ever see a positive samples. So this told us the samples, the amount of RNA we were working with was quite little in the blood. The next thing we did, after the packaging RNA tubes came out, we screened these patients. We took 226 samples, and 65 of those patients were actually, 29% of the patients were positive. And we actually did the sequencing of these gene products, and they were enterovirus. So then we had the quantitative Rt Pcr, so I said, why don't we do that? And then we figured out that, well, the levels are very low. The median was 53, and the mean was about 200. This is corresponded to our qualitative PCR. And these samples we sent to California Rickettsial Viral Disease Laboratory, that's the state laboratory, were confirmed. So I'm not going to go through the detail. Then we started thinking, how can patients have such a little amount of virus RNA in the blood, but have such a severe diffuse symptoms? And patients with hepatitis B, hepatitis C, or early HIV disease, they have anywhere from 10,000 to a million virus per ml of blood, but they have very little symptoms or no symptoms. So the answer came to us was, well, the virus must be in the tissues, as the British investigator already found in the brain, in the muscles, and heart. Maybe they went through the same thinking. So the problem with me is that we can't get brain samples, right? I mean, nobody's going to volunteer for a brain biopsy, and you don't even know where to biopsy. Definitely not the insula, right? Okay.
White House.
The White House, right? Okay. Yeah. Muscle biopsy is flawed with sampling error because you take a very small piece of muscle. You don't take a porterhouse steak type of size. And, you look at the muscle mass of the body. That's why the results were like 20, 30%. And one of the CDC expert on enteroviruses made the comment, well, that can't be the cause of this illness because it's not 80, 90%. But we sort of didn't consider the sampling error. So I said, where do we look for it? Then we realized, well, most of the patients actually have gastrointestinal symptoms since the very initial illness. Some patients have respiratory illness, then followed by gastrointestinal illness because the viruses are swallowed into the gastrointestinal. Most patients are usually referred to the GI doctor who have absolutely no knowledge of enterovirus. In fact, one of the UCLA GI doctors told one of my patients, enterovirus doesn't grow in the gastrointestinal tract. I kind of wonder, what does entero means, right? Okay. So oftentimes, gastritis was seen by endoscopy. and biopsies will show very mild chronic inflammation by histologic examination. But then people look for H. pylori, since this was discovered in 1982, and then 1997 is confirmed to be the cause of peptic ulcer disease, and Warren and Marshall won the Nobel Prize about 5-6 years ago. But the H. pylori, we only see in 1 or 2% of the biopsies. So what happened to all these other patients with inflammation? That's normal. So patients with all this debilitating gastrointestinal symptoms will get treated get referred to a psychiatrist. Okay. So stomach is actually one of the primary sites of virus replication, as we found out later. The virus should still be present if the patients still have chronic symptoms here and also have symptoms in the secondary sites, like the brain, the heart. That's the assumption. So this is a very helpful sign, and I just talked to a few patients, and I always ask them, do you have abdominal symptoms? They all do, okay? This is the stomach, the top of the triangle. This to the patient's right lower quadrant is the terminal ileum in the cecum. This is some small bowels, but mostly colon, the sigmoid colon that comes down this way. Most of the patients have 2 or 3 tender points on this triangle, 95% of the patients. So this is actually a very helpful a sign of physical examination. When patients have these tenderness, which most of them do, I can actually tell them, I think you have chronic enterovirus infection. I'll tell you the evidence why that's the case. So the stomach biopsies, this was in my first publication. On the top, you can see a lot of these brown spots. This is the immunoperoxidase showing viral protein VP1 in the parietal cells. These two are quite heavy, so we call them 2+. This one's lighter, they don't have quite as many, and that's 1+. And here is a normal one. We actually were able to block this binding with progressively increased concentration of synthetic peptide of VP1. So the summary of the first study results was 165 patients, 135 were positive, so it was 82%. And the controls, 7 out of 34, or 20% were positive. The difference was statistically significant. The enterovirus RNA was detected in 9 out of 24 specimens, that's 37%. And one of the 3 patients that had 2 specimens taken different years apart actually was positive, and only 1 out of 21 controls were positive, and the difference was statistically significant. So we also sent these biopsies to another specialist, and that was Nora Chapman and Stephen Tracy in the University of Nebraska. That was March 2006. But nothing was done about it until July 2007. 9 out of the 10 samples were found from the freezer. The 5 positive are clearly positive. 2 of them were strongly positive, and 3 are weakly positive. The 4 negative samples were negative. So we also attempted to grow these stomach biopsy samples to see if we can grow virus. Initially, we used 5-hydroxyuridine and dexamethasone-treated BGMK deaf cells. 9% were positive. And Vero 76, 14%, that's only 3 out of 22. And out of 52 specimens, 15 were positive when the stomach biopsy was growing the V30A cells for 3 days. There seems to be like a burst of virus replication, and the range of the viral RNA we detected after 3 days of growth was 368 copies to 15,000 copies. However, none of the viruses survived culture expansion. Dr. Chapman and Stephen Tracy were supposed to help us with characterizing these viruses because they already found that there's a significant deletion of the 5' end of the virus genome. Well, we couldn't even provide them the samples. This is the gene sequence of the 3D pol gene that we amplified of the stomach biopsy. They're quite similar because it's highly conserved in this area, but there's some minor differences. 5 minutes? Okay. So then we started doing the double-stranded RNA study because the basis of viral persistence has not been clearly defined. Enteroviruses are known to form double-stranded RNA in the tissue cultures, poliovirus culture, as demonstrated by David Baltimore, in the muscle biopsies of infected mice and human. So we evaluated the stem biopsies for the presence of double-stranded RNA and possible infectivity in a mouse model. So we stained this tissue, the stem biopsy here, with VP1, as you see here, and here we used the monoclonal antibody J2, which recognized the specific configuration of the double-stranded RNA. And you can see the cells are a little bit different here. And this is another patient sample, and the staining is a little bit different. So about 82% of the samples were VP1 positive, and 63% of the specimens were double-stranded RNA positive by staining. In the controls, 19% and 10% of the samples were actually positive for VP1 and double-stranded RNAs respectively. The difference was statistically significant. Then 21 out of the 23 specimens that had previously shown to have viral RNA by Rt Pcr or both grown non-cytopathic virus were also positive for the double-stranded RNA by staining. Dr. Jonas Bloomer brought up an issue with cross-reaction of 5DA/1 with other proteins, namely creatine kinase brain type sub B and also ATPase. So we actually repeated 32 slides with and without pre-incubation with antibody against creatine kinase, the C-terminus, and also antibody against ATPase, and the staining was the same. So this was published about 4 weeks ago in the Open Journal of Gastroenterology, and including this data. So then we trying to see if when we find this RNA in the tissue, is it infectious? Yes, single-strand RNA can infect a cell in the appropriate condition, but a single-strand RNA is very labile because the RNAs usually would chew it up very quickly. So, but a double-strand RNA is very hardy. It's very hard to destroy this species. So we injected 24 mice with VP1 positive, RNA positive stomach biopsies. In 2 weeks, 2 died, and 13 out of 20 of the mice had stainable viral protein in the spleen, and 1 out of 10 control specimens actually tested positive. But the control specimen were VP1 positive, RNA positive, would boil the samples, or have VP1 positive, RNA negative samples. There were 4 samples, and 2 were injected with media. However, the initial RNA and the culture results were negative, but we could have done this better. This is an example of the mouse spleen. The mouse was injected with a boiled sample. You see this nice staining here. That's all just splenic tissue when we were trying to look for VP1. And here's the one that was injected with the untreated stomach biopsy, and you can see a lot of this brown stuff. And then we magnify a little higher. You can see they're all intracellular, cytoplasmic. Okay. Okay, I better move on. So this is the very last thing. I hope I can spend a couple more minutes telling you about this case. This is important. How do we connect what we found in stomach biopsies to the brain? Well, this is a patient, actually we saw in January 2004, but before that, he developed severe vomiting and diarrhea when he was living in Japan. He had this problem for 2 months and then developed significant cognitive dysfunction, bedridden fatigue, palpitations, constant nausea, and daily postprandial vomiting. His energy level was only 2-3 out of 10. His blood work, chest X-ray, brain and spine MRI were all normal. 2D echocardiogram was normal. His laboratory studies of note is that he was Epstein-Barr virus and the HHV6 negative. This is very rare. Chlamydia pneumonia was negative. Coxsackie B virus 126 is negative. Enterovirus 11 antibody was greater than 1-640. He was started on alpha and gamma interferon. He did not feel better on this treatment, and he did not improve afterwards. Here, about 9 months later, he has such a severe stomach problem. He had an endoscopy and biopsy. You can see all this positive virus protein by BP1 staining. And here is the double-strand RNA staining we did later. Here's in the colon epithelium positive for the VP1 staining, 100x, 400x magnification. So I'm not going to go through this, but basically 6.5 years, he did not improve. And he had a lot of brainstem symptoms, and that led to this event in April 2011. He committed suicide, and his dad was kind enough to get this brain for me to help other ME patients. Initial viral studies, we took samples from 6 places, and we did not grow any virus, and we couldn't find viral RNA by Rt Pcr. A year later, when we're working with Western blot or stomach biopsies, we actually used his serum to incubate this Western blot here, and we saw these bands corresponding to about 40 to 42 kilodalton. And the 5DA, the one that's specific for the VP1, actually picked up a higher band, that's 46 kilodalton. That turned out to be the creatine kinase B, which we confirmed by using 2 antibodies in the Western blot. So we screened all this. This is a positive band for the puntomedullary junction, medulla. This is the medial temporal lobe, including amygdala. This is the frontal lobe, very, very faint band. Here is the occipital lobe, and this one is cerebellum. This is midbrain, very little. This one here is the liver, and this one is normal brain. But when we incubated with a control serum, that tested negative for VP1 binding in the EIA 15 years ago. You can see there's absolutely no binding here. This is the heavy chain of the IgG, and this is the light chain of IgG. So we went back again to do the PCR. I said, this has to be there. So when we did the Rt Pcr again, it's completely negative. There's positive control here, negative control here. Then we remember one phenomena. So we actually used the DNAs to try to digest any chromosomal DNA off this RNA. We think they are attached like this. Then repeated the Rt Pcr. Then we saw the band here for the puntomedullary junction, and this one's the frontal cortex. Here's the positive strand and the positive control and the negative control. Then we sequenced it, that band for the puntomedullary junction, okay, and that's clearly different from the positive control. And that was about 92% homology to CVV2, about 86% to Enterovirus 30. So we think this virus got inside the stomach. He was not able to contain it with his immune response. Then most likely the endovirus traveled through the vagus nerve to the brainstem by reverse axonal transport and bypassed the blood-brain barrier. Enterovirus 71 and Listeria have already been known to go through this route. From the brainstem, then the enterovirus can spread to the rest of the brain regions. This you have seen many times before. Double-strand RNA can survive in the tissue after the initial immunologic insult. This is how, like seeds of the weeds. This can open up by a viral enzyme to multiple copies of the RNA. Some is made into the protein, which demonstrated. Rarely you can see defective virus. This will turn on the immune response, which eventually interferon production and RNA cell. RNA cells will chop down the single-strand RNA, and they probably chop things into small pieces. These small pieces may bind to the DNA, where some of them could be immunostimulatory. Somehow, there's enough of these guys left. They will rehybridize, and then the viral persistence will start again. This is the model for this viral persistence. Once you have this will keep triggering interferon and the inflammatory response. And have to now acknowledge Dr. Waring, the father of this gentleman, for obtaining the brain for our study. Then all the patients who participate. Thank you.
Okay, one question. Any question? Yes, sir. One down the front here.
This is fascinating work, but could you estimate what fraction of patients around the world have a similar situation?
You mean the patient with ME?
Yes.
I think, as my humble opinion, as an infectious disease doctor seeing over 3,000 patients, the most severe cases of ME all have brain involvement. They have to have brain involvement. That's the problem with I remember Dr. Hai was arguing with me about whether, you shouldn't call it chronic fatigue syndrome. You should all be ME. But he was seeing some of the sickest patients. Yeah, I do see some patients are less ill and do not have as much brain problem. Those patients probably don't, they tend to recover much faster, just like my son. My son is quite different from one of the sickest ME patients. He did have severe brain fog, but he wasn't, you look at him, it doesn't look like he has has viruses in the brain. But eventually he recovered after taking treatments, things like that. I think this is quite common.
Okay, one more. Yeah.
I noticed that also cytomegalovirus was highly positive. Did you think that was significant in the patient?
I'll just put that question in perspective, okay? Because in 1979, 1980, when we saw the gay males come in with all sort of weird infections, we saw the first 2 cases of pneumocystis at Cedars-Sinai Medical Center. David Ho, who was subsequently the Time Magazine Man of the Year, and he's 1 year ahead of me in training. We saw these cases, and we do all these Epstein-Barr virus and CMV tests on them. It's sky high. So at UCLA, Marty Lerner was a visiting professor, actually said Epstein-Barr virus and CMV must have mutated, now are killing the T cells. 1984, HIV was found. Then CMV and EBV antibody titers are irrelevant. They're very, very high. These viruses are only relevant when the CD4 count is less than 25. Then CMV can cause invasive disease of the eyes, the lungs, the colon, and EBV can transform into a B-cell lymphoma, then where Rituxan would be used with chemotherapy. So that elevation of CMV titer, or in other patients, EBV or HHV6, has been interpreted as active invasive infection is totally erroneous. No infectious disease doctor in the United States will treat a positive high antibody titer. You have to have other evidence of disease. I provided that for you for enterovirus. Remember, I have the high titers of antibodies. Then we went to trying to find the viral protein, trying to find the viral RNA, and then we injected the stomach biopsy into an animal, and we actually killed the animal. So that's actually called evidence. I think the bar has to be set much higher when we talk about viral pathogenesis. You can't just say an elevated antibody is equals to the virus causing disease. It's not the same.
Last question, yeah.
During the 1950s, it was well recognized that young men who exercised heavily during the acute phase of their polio got very severe polio. Have you seen this phenomenon with enteroviruses as well?
Absolutely. Absolutely. The first case I saw as a fellow, infectious disease fellow at Bethesda Naval Hospital, actually Jay Sanford, who was my big boss, took me to the bedside of this Marine colonel who was suffering from myocarditis. Heart rate was 140, 150. He could barely raise his hand to shake our hands. He looked so ill, and Dr. Sanford told me this is Coxsackie myocarditis. I was just thinking, how does he know? Well, that's called experience. He has seen many cases. I've seen over 100 cases now, myself, after I start paying attention. Yeah, the 2 things he told me you don't do to this patient. Number 1, exercising. Because in animal models of enterovirus myocarditis, you inject a virus into the belly. By day 3, it reaches the heart. If you take those mice and put them through the swimming pool, every one of them would die and drown. But if you put them aside for about a month, and then let them rest, and you put them through the swimming pool again just to make sure they can swim, okay? Every one of them will go to the other side. That's been done. That's absolutely right. This is how I understood why there's this post-exertional malaise. It may not be as simple as activating the double-strand RNA in the muscles. Of course, if you have inflammation, just like our last speaker was talking about how it would influence the brain. Yeah, that probably has a lot to do with exercise. And when they're exercising too much, enteroviruses like to go to metabolically active cells, and muscle is one of the targets. So when there's a heavy infection in the muscles, there's a very large viral load in the body, and it will take forever for you to clear.
Okay, thank you very much, John.